All right, so I'm going to talk about a project that I've been working on with a couple colleagues here at UIS, utilizing drones to identify invasive species. About a year ago, we began a project exploring how to utilize drones in addressing conservation and ecological questions. Today, I'm going to share the results of some pilot work that we've carried out using a drone to capture images in the Emiquon Preserve, and then AI to identify invasive Phragmites within the imagery. So why use drones? One reason is that they are minimally intrusive. They don't leave tire tracks or paths of cut vegetation, for example. Other studies have also found that they do not cause much disturbance to wildlife. And yet another reason is that they are capable of collecting a large amount of information over a fairly large area quite quickly. So why do we need to use AI or artificial intelligence? That last plus, lots of data, can also be a minus. All of the imagery captured needs to be examined and processed. This can be a time-consuming process. It can also be quite error-prone, especially if multiple individuals are responsible for the classifications. Our solution to this problem is to train a computer model to examine the imagery and identify features of interest. And then lastly, why Phragmites? It is an invasive plant that the Nature Conservancy has been dealing with in the Emiquon Preserve. From conversations with them, we knew there were areas where we would be able to acquire large numbers of images for training the AI. Working on AI with plants is also really appealing because they don't move, so it's easy to revisit and ground truth the results the computer spits back. We also wanted to start with a single species before training the AI to recognize a larger suite of plants. In the end, there was really no reason that we had to use Phragmites to start over a different option, but it worked well for our purposes. I'm not going to spend a lot of time in this short format on our methods, but I will give a really fast overview of those things. We used a DJI Mavic 2 Pro for collecting the images, along with DJI GS Pro app for planning the flight paths. For the pilot study, we had two relatively small plots in the Emiquon Preserve. 5 times during the course of the growing season, we had the drone fly the same path and take images at the same points along the way. Approximately 60 images were captured for each plot. This is what you can see on the right. Ignore the different colored dots, but essentially the drone would fly this back and forth path and take images at each dot. These images were set with a small amount of overlap, about 10%, so we did have coverage of the whole area. In this pilot study, we flew the drone at an elevation of 5 meters. At a lower elevations like this, more detail is of course possible. However, you cannot cover as much ground on a single battery. For the pilot study, we chose to trade area coverage for greater detail. So that's what we ended up with. All images that we captured were then manually examined and Phragmites labeled. Each image was then subdivided into a little more than 1,500 patch images. These patch images were roughly 16 centimeters by 16 centimeters. Or if you don't like the metric system, that's somewhere around 6 inches by 6 inches. A balanced data set consisting of equal number of patches with Phragmites present and patch images with Phragmites absent was randomly drawn. This balanced data set consisted of around 13,000, a little more than 13,000 patch images. 70% of these were used for training, 10% for validation, and 20% for testing. So we used a little over 9,000 patch images to train the model. Following training and validation, the model was then tested using the remaining 2,600 images. Here are some examples of output. I apologize that these images are actually from a slightly different model that had a larger patch size, about 8 times larger than what we were considering in the results that I'm presenting. Even so, you can get a good feel for what the model does. The blue boxes are locations within the patches that have been identified by the computer as Phragmites. A quick look through these images and you can see some plants that this model missed. While images like these are helpful in checking on the model and catching bugs in the model, a confusion matrix, despite the name, lets us see the results a bit clearer. So we'll look at that now. So this is the confusion matrix displaying the results of the model. The target class is the real value, that's up at the top. The output class is the value as identified by the model. You can see that to the left. The red boxes are when the model incorrectly identified something, and the light blue boxes are when the model was correct. Let's focus in on the first column for right now. These are patch images without Phragmites in them. 1,159 times, the model correctly identified that Phragmites was absent. 142 times, it incorrectly identified Phragmites as being present. So we had some false positives. This could be a slight overestimate of the error. It is possible that the AI found some Phragmites that I had missed. Overall, for images without Phragmites, the model was correct about 89% of the time. In the second column, we're looking at just patch images with Phragmites. About 1,117 times, the model correctly identified patches that contain Phragmites. 184 times, the models missed the Phragmites in those images. So overall, for identifying Phragmites, it was about 86% of the time the model was correct. Taken together, the model was correct 87.5% of the time with its identifications. Again, this is essentially saying if Phragmites was present or absent in a particular patch. So it's not too challenging a scenario because we're not making multiple kinds of identifications of different plants. This is still pilot work, so we anticipate improvements as we develop a larger training set. Through the pilot work, we have identified some modifications to our setup that we believe will not only improve our results in the future, but also allow us to look at a larger area, examine where they are in a bigger landscape. We will begin work for the 2021 season in a few weeks. As part of this year's work, we will be evaluating the impact of flight elevation on the model's accuracy. We also expect to develop a larger training data set for the model, which should improve its accuracy. Our goal in the end is to be able to create a heat map of sorts with locations of Phragmites that can be used by land managers. GPS coordinates, of course, would also be available, but the map itself should provide prove more useful on highlighting areas that need further attention. So picture something like the image that we have here overlying a satellite imagery of the vegetation in the area. That is all for now. I think we have some good initial results, but we need to get back and be doing more work this spring and summer. Hopefully next year we will be able to share an update of our progress.